Hello students, how are you people? Hope you are all doing good, studying well. Right. In the last class we studied about the biomolecules and also we have discussed what is biomolecules by their chemical nature or by their molecular weight. They are bio micromolecules, bio macromolecules. Bio micromolecules means from 18 Dalton up to 800 Dalton. These are all bio micromolecules which are found in the acid soluble pool, whereas acid insoluble pool that has most of the bio macromolecules means molecular weight 800 Daltons or up to 1000 Daltons or more than 10,000 Daltons also. Well, so these are the acid insoluble pool, they can be seen in that. So lipids are also found in the acid insoluble pool, but they are again molecular weight will not cross more than 800 Dalton. That's why they are micromolecules only, bio micromolecules only, not a compulsory bio macromolecules. So the percentage wise, when we talk about the biomolecules or the chemical composition of a cell has a water content 70 to 90%. It has a protein, yes, maybe around what we can say that 10 to 15%. Okay, then it has a carbohydrate 3%, lipid 2%, ions 1%, nucleic acids 5 to 7%, what we have discussed. Fine. In today's class, we will discuss about structural units of these biomolecules. So, how these biomolecules are made up of? Let us understand one by one. So, the biomolecules and their what we can say structural units. Structural units. So, before we discuss into the details of primary, secondary structure of the proteins or anything else, yes, we need to understand these biomolecules are made up of what. Let me tell you, when we say the proteins, so proteins are mainly, yes, called as, the proteins are made up of polypeptides. Polypeptides, and these polypeptides are basically made up of a chain of amino acids. Amino acids. And then when we talk about carbohydrates, yes, these are made up of what we can say that polysaccharides. Polysaccharides. What are these polysaccharides? Polysaccharides are basically made up of, these are the chain of simple sugars. Simple sugars. And then lipids. So they are mainly made up of glycerol, what we can say that yes, they are made up of tri and diglycerides. So tri and triglycerides are again the chain of what we can say the fatty acids. They are the chain of fatty acids. And the fatty acids are basically made up of water. Yes, they are made up of glycerol molecule. Glycerols. Okay. So thus these are all what we can say mainly the basic units of biomolecules. What you are observing, proteins are basically made up of maybe more than 5,000 of proteins we can find inside a cell, and all the proteins basically made up of amino acids, and carbohydrates made up of the sugars, and mainly the lipids are made up of fatty acids and glycerol molecules. So here what I am mainly blocking, so these are the structural units of different kind of biomolecules. So as such, when we talk about amino acids, so we can say that amino acids, these are also called as building blocks. Building blocks. Building blocks of proteins. Yes, building block of proteins. So amino acids means they have the amine group and acidic group. Yes. So we can say that amino acids are having, they have alpha carbon, okay, and carbon valency is 4, means in its outer orbit carbon has the 4 electrons. So how they are shared? The 4 position occupied by, we can say that. So the basic structure of amino acid, let me tell you, basic structure of basic structure of amino acid. Yes, let me tell you. So here is the carbon. We call it the alpha carbon. Okay. The carbon has 4 valency. So it's the 4 mainly the positions are occupied by the NH2, and then here we can say that COOH, and here is the hydrogen, and here is the R. So this R is called as variable compound. So variable compound, and this is the amine group. The amine, or it is also called as the base. Then, so this is called as mainly carboxyl group. Carboxyl group is also called as acidic group. Acidic, and here is the hydrogen. So thus all the 4 positions are occupied by 4 different kind of chemicals. Okay, so the valency of carbon is 4, and all its 4 positions are occupied by, one is R variable group, other one is amine NH2 group or the base, then carboxyl group COOH, or it is the acidic group, and hydrogen. So all the mainly amino acids are same only. All the amino acids are very much similar by their structure. If the R, the variable, yes, position changes, the structure changes, and it is denoted as different amino acid. Let me tell you here, the carbon, yes, COOH minus the hydrogen, and then it is the NH2, and here a hydrogen comes. Now this amino acid is called as a glycine. Okay, yes. So in another case, the carbon NH2, and then COOH, and then here again the H, and here again, if the CH2 group comes here, then it becomes alanine. Okay, and then again let me tell you one more example. The carbon COOH minus, and here it is the H, and here it is the NH2, and if CH2OH group comes here, then it is called as a serine. Okay, so thus the variable, okay, R position is very very important. At that variable position, different compounds or chemical coming, then the structure differs. Just with the variable position, structure differs. Otherwise, all these amino acids have the common structure. What is that? Alpha carbon with the carboxyl group, amine group, and hydrogen. Only the difference occurs at R position or the variable. So this is what we have seen mainly the amino acids as such. So amino acids can be classified as, the amino acids are 3 type, or we can say they may be 4 type. So amino acids can be of 4 type. Let us say they are mainly acidic. Acidic amino acids, example, aspartic acid, then glutamic acid, then they can be, what we can say that, yes, the basic, they can be example histidine, it can be arginine, it can be lysine also, These are the few examples. Then neutral. Neutral can be, example includes for neutral amino acid, valine as well as serine. Certain amino acids exhibit kind of odor or the smell. So we call them as aromatic amino acids. So what are these aromatic amino acids? So there are number of examples. Aromatic amino acids. AA means amino acid. Example includes tryptophan, proline, phenylalanine, and it can be threonine also. It can be not threonine, it is a tyrosine. Yes, it is mainly tyrosine. These are the type of amino acids. Yes, by nature they are acidic, by nature they may be basic, by nature they may be neutral, or they may be inducing a kind of smell, also aromatic. So thus amino acids, what is the common structure? They have the carboxyl group, acidic, they have the amine group, basic, they have the hydrogen group. Then here the R group or variable group. If hydrogen, it may be glycine, CH2 alanine, CH2OH, yes it comes, then it may be serine, we can say. And then by their again chemical nature, as we said, acidic, aspartic acid and glutamic acid, basic, histidine, arginine, lysine, then neutral, valine as well as serine are the example. Aromatic compound, mainly aromatic or smell inducing, what we can say, amino acid, they may be tryptophan, phenylalanine and tyrosine also. Well, so amino acids as such, they do have one peculiar property. So what it is called as? It is called as a Zwitter ionic property. Let us understand what is this Zwitter ion and Zwitter ionic property also. So when we talk about Zwitter ions as such, so Zwitter ionic property we are going to discuss. It is actually balancing between the positive and negativity of the ions. So when we say Zwitter ionic property, Zwitter ion, Zwitter ion form of amino acids due to their what we can say that their ionizing ability of mainly the amine group NH2, and COOH groups. Well, so what happens here, let me tell you. Amino acid having its alpha carbon and having its COOH group, and it is having its H group also. Now it may have NH3 here, and it may have, what we can say, the hydrogen over here. So this is what we say, having more of the hydrogen, it is acidic amino acid. It is acidic form. Yes, it is interconvertible with one more amino acid. Yes, H, C, NH3, then COOH, we can say that. So it is COO itself, and then it is H. So it has mainly the plus as well as minus ions, and it becomes neutral. Yes, it is again interconvertible with, yes, it can be interconvertible in the form of H plus C NH2 plus COO minus and then H. So this is what we say mainly the basic. A amino acid either by losing or gaining the hydrogen ions, it can become the acidic, or it can become the basic, or it may remain the neutral. So this neutral form, maintaining its positive and negative ions equilibrium, we call it as mainly the Zwitter ionic form. So this is what we say mainly. This amino acid is a Zwitter ion form. Okay, this you need to remember properly. Fine. So I hope this point is very clear now. Let us go ahead now. The amino acids as such. Hope you may be knowing amino acids are basically 20 type. So amino acids are further classified, or the names of amino acids you need to know. Yes, they are essential as well as non-essential amino acids. I am going to give you the list of that also. So let us discuss mainly the amino acid types. So based on the cellular requirement, okay, they are essential and non-essential amino acids. Let us discuss that also. Fine. So here, as I told you, what is the functional difference between amino acids as such, essential, non-essential amino acids? When we say that they are non-essential, which are 11 by type, because they are non-essential as they are produced inside the cell only, produced within cell, so they become not much essential, non-essential. Then essential amino acids, which are 9 types. So as they are not produced in the cell, as they are not produced within the cell, need to be compensated, need to be compensated through the food we consume. So amino acids as they can be essential as well as non-essential. So let us know the list of first non-essential amino acids. So let me mention here non-essential amino acids. Okay, they can be such as the arginine, they can be asparagine, they can be aspartic acid. Okay, yes, they can be alanine. Okay, and then yes, glutamine, glycine, glutamic acid. Okay, then it can be cysteine. Okay, it can be serine, it can be tyrosine. Okay, then it can be proline. So they are all denoted by, mainly arginine is denoted as ARG, asparagine ASN, aspartic acid ASP, alanine ALA, glutamine GLN, glycine GLY, glutamic acid GLU, then cysteine CYS, serine SER, tyrosine TYR, then proline PRO. So these are what we can say that the abbreviations used to name these amino acids. Now let us understand, these are all the non-essential amino acids. Yes, they are 11 by number. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Now let us make a list of essential amino acids which are not synthesized in our body. Essential amino acids. So which are 9 in number. So they are mainly what we can say phenylalanine. Phenylalanine. They can be histidine. Okay, it can be isoleucine. It can be the leucine. Okay, it can be what we can say that mainly lysine. Okay, then it can be methionine. Okay, then it can be threonine. Okay, it can be tryptophan. It can be valine. So there are 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Phenylalanine denoted as PHE. Histidine HIS. Then isoleucine ISL. Leucine LEU. Lysine LYS. Methionine MET. Threonine THR. Tryptophan TRY. Valine VAL. So these are the names of 20 amino acids you need to remember, and the abbreviations also. This is what we talk about basically amino acids. As such, just we have discussed the alpha carbon. It has mainly the 4 positions. Valence is 4. So one side it has the carboxyl group COH. Other side it is mainly what you can say the amine group NH2. Another side is hydrogen, and the next is R variable. So the variable keep changing, and it may become the basic, acidic, or neutral also. Yes, the amino acids also exhibit due to the presence of mainly the NH2 and COH group. They also exhibit zwitter ionic form. So which is a neutral, can balance both yes, positive and negative ions. This is all what we say related with mainly what we talk amino acids. Let us go ahead now. What is that? We need to further understand the what we can say the structural units of lipids. Let us study structural units of the lipids. So now, so all we have studied about the amino acids. Let us study about structural units of the lipids, and then structural units of the sugars also we will discuss, and structural units of the nucleic acids also we will discuss. First, let us understand what is the physiological importance of the lipids as such. So first we will understand the physiological importance of the, or where do the lipids do exist and in what form they exist. Let us have a brief discussion about that. As such, number 1, so lipids mainly they form the cell membrane and membranes of the organelle. Membranes of organelle. Yes. Secondly, the lipids are long chains of hydrocarbons. But also possess the ester group COO. Then they may also have the CH2, what we say mainly the ethyl group. Even yes, they may have the methyl group also. Yes, they may have even the phosphate group also. Okay, so lipids are long chains but also possess these are the different kind of compounds. Number 3, lipids can be 16 carbon compounds. Example, palmitic acid or 20 carbon. Example, arachidonic acid we say. And then number 4, yes, mainly lipids made up of fatty acid chains containing glycerol. Okay, yes. So lipids can be existing as unsaturated with carbon to carbon double bond. Okay, or they can be saturated without double bonds. Okay, so yes, we can say that there is a little difference between saturated and unsaturated. Yes, so we need to know the difference as such. The unsaturated, okay, and then saturated lipids. So unsaturated have low melting point, they have high melting point. Yes, example for unsaturated, yes, vegetable oils, and then we say PUFA, polyunsaturated fatty acids, polyunsaturated fatty acids. So for this example, animal fat. So unsaturated fatty acids are better than the saturated fatty acids. Well, this is what we talk all about mainly. When we talk about the fatty acids, yes, the lipids basically made up of what we can say number of chemicals in that. Yes, they can be as complex. Lipids can be 7 important points. Lipids can be simple or conjugated or complex lipids. Yes, the complex lipids example, we can say that mainly the phospholipids such as lecithin, which is abundantly there in the egg yolk. So it can be sphingolipid, it can be glycolipids. So these are the few basic points related with the lipids as such by their chemical nature. Yes, we need to know all these. Yes, so we have to understand basically the every cell membrane made up of lipid bilayer only, right? And then even organelles such as it may be the mitochondria, it may be the lysosome, it may be endoplasmic reticulum, it may be Golgi complex. They are all made up of a lipid layer. Yes. So membrane bound organelle also having the lipids, and then mainly the lipids are long chain of hydrocarbons but also possess the ester groups such as COO, then CH2, then the CH3 or PO4 group, orthophosphate we say. Lipids can be 16 carbon compounds such as palmitic acid. Okay, it is a 16, and then it can be 20 carbons such as arachidonic acid. Lipids made up of fatty acids containing the glycerol also. Lipids can be unsaturated as saturated. Unsaturated having the double bond between carbon to carbon, then saturated without double bond, and then saturated mainly animal fat requires the high melting point, whereas a vegetable oil, unsaturated fatty acid, best example PUFA we can say, they require a low melting point. Lipids can be simple or it can be conjugated or complex. Complex include phospholipid. Best example is mainly lecithin, sphingolipid and glycolipids. This is what we need to know all about regarding the lipid basics. So, in the next class, yes, we will be discussing into the structural details of the different kind of lipid as well as the glucose molecules as well as the nucleic acids also. Till then, take care, study well. Thank you very much.